Hey everybody, let's go through the pathology of genetic diseases. So a lot of this will be review. Okay, so what are mutations? They're permanent changes, and it can be due to point mutations, which are substitutions of a original base with something new. It can be due to deletion or substitution. It can result in a frameshift mutation, depending on how many were lost. It's conservative if you lose just one and is replaced by the same, like if a purine is going to be replaced by another purine. That's conservative. Non-conservative is if the purine is replaced with a pyrimidine, or it could be silent, where it's like the same like reciprocal base. Nonsense if a stop codon is going to be introduced. And a frameshift mutation is if, like, it can be like 2 or 1 base is deleted. That's going to shift the frame. If there is like all 3 codons or all 3 nucleotides in a codon, that's the frame shift is going to be conservative, because it's still going through its same sequence. Autosomal dominant present late in life, and it's in every single it's going to be in every single generation. Structural proteins and receptors are mostly affected. Autosomal recessive is going to occur early in life, so it's due to the enzymes and inborn errors of metabolism. And X-linked only affects the males. Females are the carriers. And in Y-linked, only the females are affected. All of this is going to depend on penetrance, so all the signs and symptoms and the severity of the disease. Okay, so in enzyme defects, there's either the enzyme has a reduced activity or there's a reduced amount of a normal enzymes. There's always going to be a problem with feedback control. There's going to be accumulation of the substrate, and that results in the metabolic block and decreased amount of the end product. So there is another failure to inactivate a tissue damaging substrate. The example is the alpha-1 antitrypsin deficiency. Structural mutations are autosomal dominant. Well-known one is Marfan syndrome. FBN1 is going to be affected, and that's going to result in abnormal fibrillin. Problems that we need to be worried about is ectopia lentis, so retinal detachment, mitral valve prolapse, dilatation of the ascending aorta. This is a problem because it can result in a dissection of the aorta, leading to sudden death. Ehlers-Danlos is going to be a mutation affecting lysyl hydroxylase. So type 1 and type 3 collagen is going to be affected, and it's going to result in like hyperextensible skin and the hypermobile joints. Refer back to the biochemistry lectures, because we go through that in a lot more detail. Receptor protein deficiencies, most well-known one is the familial hypercholesterolemia, and that is a mutation in the gene coding for the receptor for LDL. So in the serum, there's going to be high levels of LDL. And statins don't work on families with or individuals homozygous, because there are basically no receptors. So class 1, there's going to be complete failure. Class 2, the receptor proteins are there, but they're misfolded, and they can't be transported to the cell surface, so they're just trapped inside the cell. Class 3 is when the receptor proteins are able to be transported to the cell, and they're able to bind the LDL, but they can't be like brought back into the cells. So lysosomal storage diseases are autosomal recessive. There's going to be problems with lysosomes accumulating too many products, and they interfere with normal cell function. The accumulation of the autophagic substrates and the dysfunctional mitochondria, or old mitochondria, can trigger free radical generation and apoptosis. Treat all of this by replacing the enzyme that is missing. Tay-Sachs disease is the degradation of GM2 gangliosides, and the neurons are going to be filled with cytoplasmic vacuoles, and those are going to be filled with the gangliosides. The cytoplasmic occlusions are going to be like whorled, like with a strong ring-shaped structure around it. And it's onion skin-like, so there's the little point. It's going to result in a cherry red macula due to the normal macular choroid surrounded by pale swollen ganglion. Motor incoordination is one of the main signs. So NiemannPick is the disease where lysosomal accumulation of single myelin thanks to a deficiency of sphingomyelinase. There's failure to thrive, vomiting, fever, general lymphadenopathy, there we go, which is going to be the progressive deterioration of the psychomotor function. Type A is the most severe infantile form with extensive neurologic involvement thanks to the visceral accumulation of sphingomyelin. So there's going to be progressive wasting and death by 3. It's due to a nonsense mutation. So a stop codon is introduced. The cells are going to be enlarged. They're going to be filled with sphingomyelin and cholesterol. So you can see here, look how fat, like how many how many cells are like so swollen. Like you can hardly see, like there's no nucleus in this cell. There's no nucleus in this cell. This one has one. This one has one, but they're abnormal compared to these. So they're going to be filled with the zebra bodies, which are concentric laminated myelin figures. You can't really see it too well with this. You can see it well in an electron microscope. But the type B is going to result in organomegaly, but there is no CNS involvement. So you can survive into adulthood. There are lipid phagocytic foam cells like these, and they're distributed in all these organs. And type C is going to be a mutation in NPC1 and NPC2, and that causes a defect in the non-enzymatic lipid transport. So enzyme is not going to be the problem here. Treating this with the infusion of sphingomyelinase is not going to help because the problem is the NPC1 and NPC2 transport mechanism. Gaucher's disease is an autosomal recessive mutation in the gene encoding glucocerebrosidase, and this is the most common lysosomal storage disorder. And the glucocerebrosides are going to accumulate in the phagocytes and the macrophages, and those in turn are activated to secrete interleukin 1, interleukin 6, and TNF. Type 1 is the chronic non-neuropathic form. The glucocerebrosides are going to accumulate in basically everywhere but the brain. It's going to result in splenomegaly and bone erosion, and it's found more in the European Jewish descent. Type 2 is going to be acute neuronopathic Gaucher's disease, and it's going to be an infantile acute cerebral pattern. Hepatosplenomegaly is going to occur. Progressive CNS involvement is a part of it, and there's no predilection per descendants, right? Type 3 is intermediate between these two, and it's going to be systemic and progressive CNS involvement. Gaucher disease cells are going to be found in all these organs, along with like the lymphatic or lymphoid organs, along with the lungs, so in the alveolar septi and the air cells. Gaucher cells are characterized by fibrillary cytoplasm. So these are going to have like the crumpled, like right here is a good one, tissue paper image or appearance. Period acid shift is going to be positive. And under the electron microscope, the fibrils are going to be distended lysosomes. The neurons are going to be shriveled and progressively destroyed. Mucopolysaccharidosis is going to be an autosomal recessive deficiency of enzymes that degrade the mucopolysaccharides or glycosaminoglycans. So the only exception is, not the only one, but the big exception is the Hunter's disease, which is X-linked recessive instead of autosomal recessive. And there is no clouding of the cornea. So Hunter needs to see well. in general, there are going to be coarse facial features, clouding of the cornea, joint stiffness, mental retardation, and there is going to be accumulation of mucopolysaccharides in the mononuclear phagocytic cells, the endothelial cells, smooth muscle cells, and fibroblasts. So they are also going to involve the lymphoid tissue and the heart and blood vessels. Hurler syndrome is going to be more severe than Hunter's syndrome, and the cells are going to have a clear cytoplasm, also known as balloon cells. And under the electron microscope, the cytoplasm is actually not clear. It's full of swollen lysosomes. And some of the lysosomes and the neurons are going to be replaced by the laminated zebra bodies, just like the other condition. And it's going to result in liver and spleen, megaly, skeletal deformities, valvular lesions, subendothelial arterial deposits, especially in the coronary arteries, and lesions in the brain. So here's the crumpled tissue appearance, and here are the zebra bodies. Okay, so the glycogen storage diseases, here are like von Gierke's disease, enlarged liver and kidneys, normally high levels of acid, fats, and uric acid. Pomp's disease affects all the organs. It's going to be especially the heart, so enlarged heart and liver, muscle weakness. McArdle's disease is going to affect the muscle, and there's going to be low exercise tolerance. What's going on? You can't store too much glycogen in the muscle or in the fat because it's water soluble. So you can only store it in the heart and the skeletal muscle, and those are used as like extra stores of energy. In Pomp's disease, there are no way to store the glycogen, so cardiomegaly will occur. There's the test for lactic acid, but it's going to be low because there's no glycogen to be converted to lactic acid. Okay, genetics. Proto-oncogenes are regulating and stimulating normal cell growth, and they're regulated by the tumor suppressor genes. So clearly, anything wrong with these two, there's going to be cancer. Neurofibromatosis type 1 is going to be when NF1 gene is mutated, and there's going to be a neoplasm of the Schwann cells, the fibroblasts, and neurofibromin. Results in the cafe au lait spots, neurofibromas, and Lesch nodules. These are your Lesch nodules. Choristoma heterotopia is when a wrong type of tissue there's wrong tissue type in a location. So, let's say there is like Merkel's diverticulum. So in the intestine, it should be intestinal tissue, but in Merkel's diverticulum, there's going to be pancreatic tissue there. Neurofibromatosis type 2 is a Maryland mutation, and there's going to be bilateral acoustic schwannoma. So here it is. There's a cancer or tumor. It's going to compress on the acoustic nerve, and it's going to be made out of the Schwann cells. The Schwann cells are going to enlarge, compress, and result in cortical deafness. Chromosomal disorders tend to be autosomal dominant. Aneuploidy is usually caused by non-disjunction or anaphase lag, and mosaicism is caused by mitotic error during the cleavage of the fertilized ovum or in the somatic cells. So the interstitial deletion is going to result in the no centromeres, and ring centromeres are going to occur. So an inversion involving one chromosome arm is paracentric, and if the opposite sides of the centromere is going to be affected, it's pericentric. Inversions aren't too bad. They're compatible with normal development. Isochromosome 1 is when, or isochromosome is when an arm is lost and the remaining arm is duplicated. So the most common one is XQ isochromosome. And translocation could be balanced, reciprocal, and these people are phenotypically normal, but when their child is produced, that can be a lot more problematic. It can result in like trinucleotide repeats. A Robertsonian translocation is a translocation between two acrocentric chromosomes. So these are going to break and rejoin, makes just like one big chromosome, and the little fragment is lost. It's compatible with life, phenotypically normal, but problems occur when these people are trying to conceive. Trisomy 21 Down syndrome is caused by a Robertsonian translocation or mosaicism. Most important problems we got to look out for that is the endocardial cushion defects. So there's going to be either like atrial septal defect, ventricular septal defects, cancer we have to be worried about. So in particular, megakaryoblast leukemia. All of them will develop Alzheimer, and most of them have some kind of T cell deficiency affecting their immune system. Chromosome 22 Q11.2 deletion. So it's a deletion on the Q arm. There's a spectrum of disorders. Famous one is DiGeorge syndrome, which is thymus hypoplasia. Okay, so Klinefelter's is going to be 47 chromosomes. XXY is due to non-disjunction during meiosis. So there's a genetic male, but he's not going to form secondary male characteristic. Risk that we have to be considering is the mitral prolapse, osteoporosis, and breast cancer, because there's no estrogen to prevent these, right? Well, the osteoporosis and the breast cancer. Infertility, because the testicular tubules are atrophied and they're going to be replaced by pink hyaline or is underdeveloped. Prominent Leydig cells, feminine physique, mildly impaired IQ. Turner syndrome is going to be 45 chromosomes. It's just one X chromosome. And there is complete or partial monosopy, so there could be like a little arm. But it's the most common sex chromosome abnormality in the females. And there is going to be a cystic hygroma that forms, usually in the neck, so it looks like there's webbing. And there's coarctation of the aorta, so this is the one that you need to be worried about because that can lead to heart problems in the future. This person is not going to have secondary sex characteristics, so there's no breast formation. There's also an arm deformation that it's in the elbow. There is, it's the most important or the most common cause of primary amenorrhea. Usually, this is not really detected in early life. People come in, well, their parents bring the child in after 16 something, and they say like, hey, my child hasn't had a period yet. So you got to consider that, hey, this person might have Turner syndrome. Other things we need to consider, they can be hypothyroid conditions, glucose intolerance, obesity, thanks to the insulin resistance. But you can't give growth hormones with insulin, it's contraindicated. Streak ovaries are going to result in the early menopause because the ovaries haven't really developed. As you can see here, there's just a streak. It should be like a egg shape. So their ovums are not formed, and the developing eggs are usually destroyed before the person is even able to menstruate. Because remember, there are many eggs that are formed during development, but you don't actually end up with many when you actually reach puberty because a lot of them degenerate. Trinucleotide repeats. One thing you had to know about is the Fragile X. It's the second most common genetic cause of mental retardation after Down syndrome. It's a mutation in the familial retardation gene. Horrible name, I know, but that's the way it is. FMR1 selectively binds mRNA associated with polysomes and regulates the intracellular transport to the dendrites. Another disease is polyglutamine disease, and is progressive neurodegeneration in the midlife because of a gain of function, so too much glutamine. It's going to accumulate in the intranuclear inclusions. Fragile X tremor ataxia is thanks to CGG premutation that can progress to Parkinson's. The FMR1 gene is not silenced, it's transcribed and accumulates in the nuclei. Here's the nucleus. This should be the only thing in it, but look, there's new stuff. extra stuff in it. And that forms the intranuclear inclusions. Leber hereditary optic neuropathy is a neurodegenerative disease that manifests as bilateral loss of central vision. So here you can see there's thickening of the peripapillary RNFL, and it's pale in the center. So right here, this is the vision of normal. This is a person suffering from LHON, loss of central vision. Genomic imprinting, the ones that you need to know about are Prader-Willi. It's due to deletion of the paternal contribution of chromosome 15, and affects women. So there's going to be mental retardation, a short stature, hypotomia, hypophagia, but most importantly, obesity. This obesity is going to occur in children, and they're going to be morbidly obese ever since they were like 5. Angelman is going to be the deletion of chromosome 15 from the mother contribution, and ubiquitin ligase is going to occur. It's also known as happy puppet syndrome because the child will always be smiling. They also can have seizures, ataxia, retardation, inappropriate laughter. There's actually problems with the vestibular system in Angelman syndrome, so it always kind of feels like to them that they're on a roller coaster. That's why they're always like laughing and giggling and kind of like shaking, because it might not be an actual seizure. That's it for now. Thank you for your attention and good luck studying.